All right, everyone. Welcome back to Biology, Intro to Biology. This is chapter 13, Evidence of Evolution. Now, last time we talked about chapter 12, which is all about what causes evolution to happen. What are the forces? Genetic drift. It could be the environment. It could be different proteins that are being exhibited in an organism through some random mutation. It could be any one of those. But remember, evolution does not occur unless there is reproductive success. As long as that organism can reproduce successfully and produce multiple offspring with that exact same phenotype, remember what phenotype is, then and only then does the process of adaptation to evolution can occur. And that's usually over a period of time. The whole population will change into that result. So just a little tip before, not a little tip, but just a little announcement before we even get started with chapter 13. Some of you guys have emailed me and notified me that last week's lecture, the sound was cut off. And I apologize for that, guys. I did not know. It could have been with the upload of YouTube. I did have trouble uploading it to YouTube. It was stuck at a certain percentage for a good 20-30 minutes, and that could be the reason why. Maybe the file's too large. I don't know. So if there is another audio clipping here, please let me know so I can re-record it. I will be re-recording the sections that was clipped out in chapter 12 for you guys, and I'm going to post it up later this week. Your exam is coming up next week. So just to recap, and this is a very rough recap because we're no longer face to face, so we can't really do this in person, but I'm going to do it with you guys right here. Chapter 8 or chapter 9, can't remember which one was, meiosis. Again, if you guys can start seeing the patterns of how these chapters are organized, you'll see that meiosis, what's the whole point of meiosis? There's 8 stages. Don't forget the 8 stages. But the whole point of meiosis is to create gametic cells. Now, each of the gametic cells are uniquely created. So they're not identical. They're all unique cells. And depending on those unique cells, once it becomes fertilized, then it becomes an organism. And depending on what's inside each of the gamete cells, different alleles are being passed down to the offspring. That goes into the next chapter, which is allele distribution, during random distribution, frequency distribution, what have you. That's all there to help condense and concise. Not condense and concise, but it's there as a way to pass on genetic trait. And so we did have a chapter, I believe it was chapter 11, that we calculated what were the, what do you call it? What were the with the Punnett square, so to speak. So we did use a Punnett square. We had mom and dad's genetics, alleles, and then we crossbreed them to see the probability of the offspring. Okay, so I believe that was chapter 11. Chapter 12, or not chapter 12, but the animal development is once those gametic cells are fertilized, how does an embryo form? Okay, so that's now on the process of embryo development. Once the embryo is developed and turns into an organism, what are the forces that causes a population in general over time to change and to adapt and to evolve? That was the last chapter. So this chapter is how do we know evolution actually existed, and this is the whole theory of evolution. So let's just get started right now. I believe, again, your exam's based on 4 chapters, so I believe it was chapters 9, 10, 11, no, 9, 10, 12, and animal development. Again, in the syllabus, look at it. It should be based on 4 chapters. I don't believe the chapter 13 is on it, but if it is, if it's written in the syllabus, it's on it. So hopefully we can get this out of the way for next week. And next Monday, we might... Nah, forget it. Later. Okay, so let's get started. Chapter 13. All right, let me get started here. All right, so Earth was created 3.8 billion years ago. And don't ask me how this happened, how this worked. I have no clue. But we do have tools today that helps us guesstimate. And I use that term, guesstimate, because we don't really truly, at least from my understanding, we don't truly understand how far back the Earth goes. So all we know is that we have a geological time scale. And all this means is that Earth has been divided by different areas of mass extinction and mass population. And over time, you're going to see layers and layers of sediments throughout a rock formation. Now, the older the sediments are, the more lower it is on the rocks, and the newer it is, so to speak, the more on top of the layer it is, or the more surface it is on the layers. And so there are multiple different eons and eras. You can see right here on the bottom right hand corner, we had the Precambrian era, we had the Archaic era, we had the Mesozoic era, you know, and you're gonna have the Triassic period, Jurassic period, all found within these time frames. So this is just an introductory slide. Let's keep going. Again, all we know is the evidence that's left behind after the extinction occurred from way back when, and we see this quite often, especially in the rivers and streams. If you were to dig into deep within a mine, you might find some fossils, and these are just fossilized organisms. Either they're plants, crustaceans, or even animals, bones, even dinosaurs. And the whole process of studying fossils is what we call paleontology. So paleontology is not only the study of dinosaurs. I think that's another terminology for it, but I've always connected the two with the same word. But paleontology is the study of fossils in general. And it's these fossils, especially where they're found and how we can actually date them, meaning guess how old they are, that gives us an idea, a rough idea of what organisms lived during that era of sediment. Sorry. Okay, so here's some randomized fossils, not randomized fossils, but here are some fossils that you guys can see. This is an extinct plant right here. Actually, let me use my pointer. Haha, laser pointer. Okay, we have a random plant right here. I don't know what the plant is. We have a Triceratops right here, and then we have some crustacean over here. This looks like a snake-like creature or fossilized amber or something like that. We have fossilized wood. Believe it or not, there is fossilized wood. It's very hard. It has. I've actually had fossilized wood before. Someone gave it to me. I don't have it with me anymore, but it is a very hard-like structure. It's very brittle too. It breaks off very easily, but it doesn't have that wood quality. You know, when you touch wood and it has that feel to it, that nice wood feel, not plywood, but just regular wood. If it doesn't even feel like that, it feels more like sandstone if you really want to think about it. And then right here, it looks like a fossilized bird, so to speak. So again, this is how organisms are preserved. Now, if you know of the Jurassic Park, if you've seen the movie Jurassic Park, some organisms can be fossilized in amber as well, which is just fossilized tree sap. And you know that little mosquito on the staff of I forgot what the actor's name was. So something Attenborough. It's David Attenborough's older brother. But that little mosquito that's stuck inside that amber is a fossilized mosquito. So what is the geological time scale? Again, break the words down. If you see a word that you don't know on the exam, break the words down. What does geo mean? Logical? Logic? Again, biology? Logic? Logi? The study of. So geo, earth. Logic, study of, pertaining to the study of Earth, timescale. Earth's timescale, so to speak. So there you go. So even though fossil evidence is diverse, it is often challenging. And the reason why it's challenging because there's so many fossils everywhere. And sometimes the fossil is not preserved all the way. Sometimes we only have pieces of fossils. The Earth's plates are always moving. Remember, we live on tectonic plates, so there's a constant movement of the Earth's surface. Mountains are being pushed up by two tectonic plates hitting each other, and then there's also valleys and ridges that form underneath it. And so sometimes the plates are just going to overlap each other, and it's going to get buried even further deep into the Earth's crust. So this is just one of the examples of how it's hard to base this whole theory on fossils, but again, this is what we do. Okay, so let's keep going. Come on. Why is this not moving? There we go. So another way to learn how to date the Earth's age is by using a process called relative dating. Essentially, in order to find out approximately how old the Earth is, we have to look at radioactive isotopes. Now, if you remember all the way back in chapter 2 when we talked about atoms and what atoms are made of, remember that we're working with isotopes here. Now, isotopes just means it's an atom with a different number of neutrons in it. And depending on how much neutrons we shove into that nucleus, that atom becomes more and more unstable. And because they become more unstable, chances are over time, they're most likely going to break down and degrade out. So that's why we like to use radioactive isotopes. We also use isotopes in medicine, but that's getting off topic and that's for another lecture. So let's come back. All right. So there's two ways to do relative dating. There's absolute dating, or just dating in general. Absolute dating. So absolute dating uses chemistry to determine how long ago the fossil was formed. So we'll talk more into that about that, but let me read the second term. So radiometric dating is a type of absolute dating that uses the radioactive isotope, which is what I'm talking about here. So if we want to know how old an organism is, we look at different isotopes. So sometimes we'll look at C14. Sometimes we'll look at N14. That just means carbon with 14 nitrogens stuck in the center of the atom, or nitrogen with 14 neutrons. Did I say nitrogens? Neutrons, sorry. The 14 represents the number of neutrons stuck in the atom, in the atom's nucleus. Okay? So how this concept works is that we are made out of carbon, nitrogen, and oxygen, and hydrogen, mainly. That's what makes up most of it. That's what we call an organic molecule. If you guys are going to take organic chemistry later on, you are going to be so familiar with that, with those four atoms. Anyways, so the concept is, as our body is developed, sometimes we take in radioactive isotopes. And this occurs naturally in nature, not on a huge abundant scale, but it does occur. And within us, we have a certain number of radioactive isotopes. So what happens over time as our cells die off and as we die, there are no new carbon-12 or carbon-14s added. Again, we're not living, we're not dividing, we're not absorbing any new nutrients. So they are going to slowly leave the organisms over time. Sorry, that's my phone. And they're going to slowly leave the organism over time. And as they leave, they then become, so to speak, we're able, not become, but we're able to calculate how much of that carbon-14 or nitrogen-14 atoms are left within our bones. And if we understand the half-life, which is what's being shown right here on this imagery, if we understand the half-life of each of the carbon or nitrogen isotopes, we can then back calculate or backtrack it so that we can find out how old essentially the organism is. And again, we're going to do an example so that makes a little bit more sense. If you still don't understand it, just go on YouTube. Really, there's a lot of great YouTube videos out there. I can't do it because of copyright, but Google radioactive dating on YouTube or for biology, and that should pop up a better explanation if my explanation was a little how do I say it confusing. So anyways, so as we look right here, as an organism is alive, carbon-14 and nitrogen-14 is somehow interacting with each other. So carbon-14 leaves as nitrogen-14. Interesting. Again, with radioactive material, the neutrons are going to play a pivotal role in this process. Okay, I don't know too much about it. I'm going to be honest with you guys. I don't know too much about radioactive dating. So That's why I'm recommending you guys look it up, because this is something that just flies over my head. I understand the calculation, but the concept of it is still kind of vague to me. So I'm just being honest with you guys, all right? Now, if we look right here, throughout the organism's life, there is going to be an accumulation of carbon-14, and this is radioactive. So as we are living, we're going to have a constant supply of that carbon-14. Again, there's not an abundant amount in the world, and if anything, they're evenly distributed amongst all organisms. So as long as we know that, and we know how long it takes for half of those carbon-14s to disappear from our bodies, we then can backtrack how old the fossil is. So right here, the half-life of carbon-14 is approximately 5,730 years to get rid of half. So if we started out with 10 carbon-14 molecules or atoms, in 5,730 years, only 5 will be left in our example. That's how long it takes. And then another 5,730 years, there will only be 2.5 atoms left. It gets cut in half every single time. Again, this is how half-life works. Okay? So, after the organism dies, no more carbon-12 or carbon-14 is added. However, carbon-14 decays at a constant rate, leaving the organism as nitrogen. There we go. It helps explain us. I don't understand the microchemistry to that process, but just know carbon-14 will leave as nitrogen in the very end. Okay, so a proton somehow ejected out, and nitrogen is left. Okay, so carbon-14 decays at a constant rate, leaving the organism as nitrogen. Okay, we just read that. So right here, during any 5,730 year period, the amount of carbon-14 is always cut in half. Again, this is essentially what I've been telling you guys this whole time. So if I was to ask you guys a question, which rock layer, A, B, C, or C, should have fossils with the most carbon-14? Now, really think about this question. Which rock layer should have the most carbon-14? So I'm going to give you guys a minute. Pause this video and just think about it. Okay. I'm assuming that you paused it. Go back. Come on. All right. I trust you. All right. What's the answer? If you guys said C, I'm sorry, that is the wrong answer. Because C, if the question was asking which rock layer is the oldest, C would be the correct answer. And the reason why is because it's deeper in the layers. This A is where the surface is, and C is the lowest layer, so it's known to be the oldest out of A, B, and C. Okay. Now, when we're asking for the most carbon-14, now remember, we have a sustained amount of carbon-14 as we are alive. Okay. So that means the longer the time has passed after our deaths, the less carbon-14 we have in the fossil. So essentially, more carbon-14 means we're more alive. Less carbon-14 means we're more dead. We died a longer time ago. English, go figure. So the answer would be A, guys. A will have the most because it is the newest layer to have formed. Okay. Now, researchers used a radioactive isotope with a 25,000 year half-life to date a fossil to around 100,000 years ago. The fossil contains blank as much of the isotopes as does a living organism. Fill in the blank. Now we're looking for a percentage here. Okay, we're looking at rough approximation. Remember, half-life, which is the key word in this problem, represents 50%, and half of a half-life, or half of 50%, is another 25%. If you guys don't understand that too well, grab a calculator, divide it by 2. Take 100 divided by 2. What percent are you left with? 50%. Divide that by 2 again. What percent are you left with? 25%. Okay? So, take a moment, work this question out. This is a great question, especially for multiple choice. work this question out and see what you get. Okay, you're back. We're gonna work through this together. I really wish I had a pen and stuff. I only have a mouse. I still don't have a pen. Amazon, especially during this time, is not shipping stuff as fast as it used to. So bear with me. Hopefully by exam 4 we'll have a pen. So anyways, let's get to it. So I'm going to use my pen here. Okay, now the key word is half-life. So I know this is a half-life problem. I know that every half-life takes 25,000 years. We dated this fossil to be 100,000 years old, because this says to date the fossil to 100,000 years ago. So with that being said, I know that the time at 100,000 years ago, so here's present. Okay, this is the present day. Oh my gosh, I can't write. Okay, so I know to the present day, this time frame right here is approximately 100,000 years old. Okay, and I know that at the beginning of the death, this is the death. Okay, this is the death time right here. I know at the beginning of the death, we had 100% carbon-14. Again, remember, we have 100% when they are alive, carbon-14. It is only when they die. The moment they die, that's when the half-life starts counting. So if we're at 100% when they're alive, once they die, that's when the percentage starts going down. Okay? So if I know we're at 100% at death, right at the moment of death, and the half-life, which I'm just going to label as 1/2 L, equals 25,000, I know for every 25,000 years, okay, 1/2 of the area is done. So the question is, how many half-lives can fit in 100,000 years? Well, I know that every half-life is 25,000 years long. I know this whole span right here is 100,000. So if I take my calculation, do 100,000, or I'm just going to say 100,000, okay, divide that by 25,000. Again, I'm writing with a mouse. This will give me approximately 4 half-lives. 4. So that means that I'm going to divide this about 4 times. Okay, so I know I'm at 100% right here. The first half-life, I'm now down to 50% because this is the first half-life. The second half-life, I'm down to 25%. Okay, because that's the second one. The third half-life, I'm down to 12.5%. Yes, 12.5%. So right when we hit that fourth half-life mark, 12.5%, divide that by 2. Again, every time we do a half-life, we're dividing by 2. 12.5 divided by 2. Let me get my calculator. I don't have a calculator, so I'm going to use my phone. So what is 12.5 divided by 2? I can't do math right now. So 12.5 divided by 2 is 6.25. Okay? So 6.25%. So that organism that that the fossil we have found will contain 6.25% as much of the isotopes as it does for any living organism. That's the answer. So, which is 1/16. So if I did math, 1 right here, let's see, 1 divided by 16 gives me 0.0625. Okay? 0.0625. And if I multiply that by 100 or move the decimal place over 2 places to the right, that is percentage. 6.25%. Okay? So I did it one way. They did it a fractional way. Let me just show you the fractional way really quickly. Let me erase this really quick. Okay. Dum, dum, dum. So right here, pen. So they did it with fractions. So in order to show a fraction for 100%, we do 1 out of 1. Okay, and we know that it took 4 half-lives. Okay, so we're going to multiply it by 4 half-lives. 1 over 2. Okay, times it by another 1 over 2. Times it by another 1 over 2. Times it by one last 1 over 2. Okay, and if we were to take this whole value, multiply straight across, that's 1 over straight on the bottom, that's 16. And that is my answer, 1 out of 16. Okay, that's how they did it. But you would have to understand what we just did earlier, which is we have a total year of 100,000. We have a half-life of 25,000. So how many times can 25,000 fit in 100,000? That's 4. Okay. Now distinguish between relative and absolute dating of fossil. Remember, absolute is using precise chemistry to calculate the age of a fossil. Relative dating is guessing between the fossils, the era of the rocks, and all of that, so to speak. Okay, so Earth's geography moves. Okay, originally, back when the Earth was first forming, or was first there, we have a supercontinent called Pangaea. Now, if you look, Pangaea looks like a big conglomerate of land, just one land. This is present day South America, North America down here. And you can see that South America has this nice, almost puzzle-like fit to Africa. And you can see that it's theorized that South Africa and Africa was once a supercontinent back during this era right here. I don't know what MYA stands for, so I'm not going to say it. Million years ago. There we go. That's what it stands for. MYA means million years ago. Oh, science. Anyways, so Pangaea was a whole supercontinent. Now, why do continents move? Again, Earth is a moving landmass. We are positioned on plate tectonics, which is floating on top of magma. And when you see a volcano erupt, what pours out of a volcano? Lava, which is magma. It's just magma. Once it pierces the Earth's surface, is called lava. Whatever. Anyways, so we're floating on piles of magma. So it's like having ice cubes inside your cup, and it's just moving around. That's how our continent moves, but at such a slow pace because they're extremely heavy. It takes a lot of energy just to move them a couple inches each year. So, and when they do move, that's when earthquakes occur. Okay, so again, plate tectonic theory. We're living on top of movable plates. And the reason why we theorize that we lived on a supercontinent called Pangaea is because fossils were found throughout these continents. Seemingly, for some reason, even though today they're completely separated, we're able to find fossils, this Triassic land reptile, in the regions of South America and Africa, as well as this Lystrosaurus, which is another land reptile, in Africa and India, and as well as Antarctica. The only way that we were able to find that species in that whole range is if they were connected together. Because in today's world, they're not going to swim across the Atlantic Ocean. I mean, it takes a long time. And I don't think we're going to find that many fossils along those different regions, especially in Antarctica today, where Antarctica is in the South Pole. Like, the temperature there is below 0 degrees. Versus Africa and India, it's like a hot 90 degrees Fahrenheit. I don't know what the Celsius temperature. When I said 0 degrees, I meant Celsius for Antarctica. Below 31, 32 degrees Fahrenheit for Antarctica. So anyways, we're finding fossils everywhere in different environments, in different heats, temperatures, latitudes, and everything. The best explanation that we can come up with is if everything was shoved together into one gigantic landmass. So, this is the study that was done. And again, we see a lot of, how do I put it? We see a difference in fauna, so to speak, or a difference in land mammals or land animals along this Wallace line right here. For some reason, to the left of it, we're able to see a lot of rhinos, elephants, orangutans, uh, just a lot of those species involved in fossilized too. And to the right of that, for some reason, we're seeing a lot of kangaroos, honeysuckers, brush turkeys, and sugar gliders, and all of that. So there seems to be a very distinct line where organisms just somehow evolved independently from each other. There is no, how do I put it? There is no influence on their evolutionary state. You can see that the type of animals, especially a rhinoceros, compared to a deer, is vastly different. So there is some variety for each of this. Okay, so how have the positions of the Earth's continent changed over the past 200 million years? Pangea, guys. Okay, so here's the thing. We know that during animal development, which is the sections that I lectured a little bit earlier than this, we know that we have very similar development, especially during the embryonic stage. You can see that we're almost nearly identical. And because of that similarity, and because we do share our DNA, human DNA, we are 80% similar to that of a banana, or is it 60%? We're 60% banana. We share the same DNA to a banana. I believe it was like 80% to a fruit fly, 99.9% to a chimpanzee, our DNA itself. So there's that only 1% different. So all organisms, we share DNA. And because of that similarity, we also develop similar limbs or similar functions of limbs. And so when we see a very similar type of limb or a similar makeup or buildup of the limb itself, this is what we call a homologous structure. It's just a structure that's very similar. It's probably because we came from a common ancestor. And so here are some examples of animals that have very similar bone structure as us. I forget. I know it's the radial bone is this bone right here with the brachialis, and I forget what the big bone is called up on top. I know the femur is the leg version, but you guys get the idea. Our arms, and you can see there's multiple bones in the digits, is very similar to that of a seal and of a lion. You can see that the lion has multiple digits right here. They do have that brachialis right here as well as the radial bone. Same thing with the seal, brachialis, radial bone, and then I forgot which is one this one is called. Even a bat has very similar function right here, brachialis, or that's radial bone, and on the back of that is a brachialis with the same. So we all, it's theorized that we come from a common ancestor. Long story short. Okay, there you go. So we circled it right here. Okay. Now, vestigial structures are essentially structures that have been lost over time. Some animals, especially during the evolution, during where the environment is, how they grew up in that environment, sometimes it's just extraneous for them to have that extra limb. They don't need it. It's a waste of energy. Remember, everything's about efficiency in biology. From protein development to DNA replication to cellular replication, everything is about energy efficiency. We don't want to waste energy for a structure that we don't really need, so to speak. Okay, so most structures, especially in this example, they lost their function. Our appendix, so to speak, is a homologous structure to a lot of organisms. You know, it's a homologous organ to organisms, but we don't really use our appendix anymore. That might be a bad example, but in this case, you can see a snake no longer has legs and no longer has any fins, so to speak. So they were able to evolve over time because that is what best suits in their environment. Now, granted, back then, if you look at a bone of a skeleton, you can see tiny little limbs, tiny little leg limbs and arm limbs in their skeletal structure, which represents the idea that, hey, look, they used to have it, but over time they lost it. And this is just a representation, graphical representation of one of the extinct fossils where you can actually see these slender like reptilians. It looks like it's also aquatic due to the webbing of the imagery right here, but they did have limbs back then. Okay, so anatomical structures that are analogous, okay, they are similar to each other but did not come from the same ancestor. For instance, you know what? I think there is an example after this, so let's hold off on that. None of these cave animals has pigments or eyes. Oh, there's the example right there. See, I'm not reading the slide. I'm actually doing it off of memory. I'm not reading the slide. So if you look, these animals right here do not have any color in their skin, and they don't develop eyes, but they are not related whatsoever from a recent ancestor. Recent, okay? We might have been all derived from one common ancestor, but if we're looking at a tick versus a salamander versus a crustacean, there is no recent ancestor that we can see that they come from. It's not like a crawdad with a lobster. Like, those are very similar. and so it's nothing like that, but they all have the same adaptation: no eyes and pale skin. Now, these are cave animals. So cave animals, look in the environment. The cave does not have a lot of light in it, hence that's why it's called a cave, guys. But it doesn't have a lot of light in it. And so if there's no light and nothing can see in the dark, does it really need to spend energy to develop eyeballs if there's no light? Probably not. Does it really need to show a display of beautiful colors to attract a mate? Probably not, because the mate cannot see it. So these are just adaptations that have occurred because there's no point in spending excess energies if you can't even utilize it. So, excuse me. So that's an example of an analogous structure. And these arose by convergent evolution, which produces similar structure in an organism, but they don't share any lineage to each other whatsoever. Again, what we just talked about. Okay, so the streamlined shape of dolphins and sharks evolved independently. Now imagine, what does a dolphin look like? What does a shark look like? Okay, now the body plan of these two animals are what? Homologous? Vestigial? Analogous? A product of convergent? Or both C and D? So take a moment. I'm going to give you guys a moment. Okay, let's see what the answer is. They're both C and D. They're analogous to each other because, again, the environment, they're both aquatic, right? In order to move, submerge through a water, you need some stabilizers on you. Hence the fins, the dorsal fin and the two side fins. And it's also the product of convergent because the dolphin and a shark does not share a common ancestry. If anything, a shark is a fish. A dolphin is a mammal. If you guys can't tell, the tails. Look at the tails. If the tails are flat and they go up and down, that's a mammal. That's how a dog runs. If you see a dog running or a cat running, you can see that their hind legs and their tail goes up and down. Excuse me. I gotta close my window. Okay, I'm back. Sorry about that. It's trash day. So, if you look at a fish, fish will wave its body back and forth to shoot through the water. Again, that's their evolutionary trait. So that's how you tell the difference between a shark and a dolphin. Next. What can homologies reveal about evolution? Take your time. Okay, we're just gonna keep going. So Here is a development of a fetus, chimpanzee, and human, as well as an adult chimpanzee and human. Anatomically, we are very, very similar. And if you look at the embryo development, we're almost nearly identical. I mean, we do share 99.8% of our DNA together. But look at the skull structure here. Okay, what do you notice? In terms of the development of a baby's skull, they are almost identical. Like there's almost no difference. You might be able to see a bigger jawline right here. The developmental of the skull. Remember, baby's skull is very soft and very malleable, so you don't want to push on a baby's baby's head unless you have a little dent in there. But as you can see, as they grow older, the big feature that's the difference between us is the teeth. You can see the jawline of a chimpanzee is much more pronounced, much stronger, as well as the size of the cranium or the size of the skull. You can tell that we've evolved to use more of our brain, whereas the chimpanzee's skull is much softer or much smaller. Okay, that's how we're different. Again, this goes back to animal development. Look at the development of each of these organisms. Homeotic genes. Again, homeotic genes is just the genes that is in effect during the embryonic stages. You can see that the yolk sac, so to speak, is found on most of us. This is an alligator, a mouse, and a fish. You can see the head starts developing right here, same as here, and same as the alligator, and then the tail. Very similar, very, very similar. Okay. Again, it's the genes that control the developmental stages. Excuse me. And this. Okay, so homeotic genes, again, sorry about that. I had to pause just really quickly because there's a lot of noise in the background. But homeotic genes, depending on how much of the gene is being expressed, will determine if you develop certain phenotypic traits. For instance, in a baby chicken, these homeotic genes only overlap in these two regions right here. So this region right here, okay? But because of that, forelimbs will then begin to develop as well as hind limbs. But if you look at the homeotic genes right here, they take up a lot of more space where there is no more limbs developing. Okay, and this is the homeotic genes of a python. So how does study of embryonic development reveal clues to a shared evolutionary history? Again, this is all chapter, not even chapter, the animal development lecture. So look over that. Okay, and again, if you compare the DNA and protein sequence, it's highly unlikely that two unrelated species will evolve precisely using the exact same DNA. There's always going to be variation. There's always going to be different expressions of different proteins. And because over time mutation does occur, our cells mutate every single day. Some of it doesn't harm us. It's a silent mutation, whereas others is more harmful. And our cells will undergo apoptosis if things get out of control. And when it doesn't get out of control, what does that become, guys? Cancer. Okay, so cytochrome C is a mitochondrial protein that is often used in molecular comparison. It's just a protein marker. This is what we can use in Ancestry.com. This is what we use in 23andMe. We use protein markers to see our relationship between each other. This is how genetic analysis works. So, number of different amino acids. Difference from humans, chimpanzees 0, we're pretty identical. 1, 9, 10, 12, 20, up to 42, depending on which organisms you're looking at. Okay. Again, there's a molecular clock assigned to dates to evolutionary track. If a gene is estimated to mutate once every 25 million years, then two differences from an ancestor might arise in 50 million years. Very similar to the concept of half-life, except now we're dealing with a more genetic mutation. Okay, so So if a gene is estimated to mutate once every 25 million years, that is the mutation time, not half-life, mutation time, then the difference from an ancestor might arise in 50 million years. Now, it could be earlier than that, depending on what type of mutation it is, but for this example, after 50 million years, we might have 2 mutations occurring. Okay? And that will be a rise to 1 modern species. And because it's different, and so the other organism might approach the different pathway, that arise to the next organism. But the common ancestor is the main ancestor right here that both of these species share. The only difference is these two what do you call this? See, I'm blanking out too, guys. This happens all the time. Base pair. Jeez. The base pairs are different, but we can track them back to one unique DNA, common ancestor. Okay? So a mutation in a gene occurs at a rate of 1 nucleotide every 10 million years. The gene sequence differs by 6 nucleotides, only 6, between 2 related organisms. How long ago did these organisms split from a common ancestor? Take your time. Come back to me when you're ready. Okay, you're back. So, mutations. Always ask yourself, when dealing with a world problem, ask yourself, what is the question asking? This question is asking, how long ago did the organism split from a common ancestor? So we're trying to find how long ago was this common ancestor. So we're trying to find this. This is what we're trying to find. This is species 1. Okay. Species number 1. This right here is species number 2. Again, I'm writing with a mouse, so this is very messy. So we're trying to find out how far back it goes. Well, we know that the difference between these two species is 6 nucleotides. That's what it says. Okay, I'm just going to write NuCl. And every mutation occurs every 10 million years. Since they're different by 6, what does that become? Okay, 30 million years. Now, why is it 30 million years? Because to go from a common ancestor to here, we have 3 nucleotides that will change. Not 6. Just bear with it. 3 for now. Okay? Because every 10 million years, 1 nucleotide per path is changing. So a better representation right here would be this. Okay? Let me get it here. Pen. So say that I divide this whole pathway into 3 pieces. Now each pathway, depending on the path, will mutate 1 nucleotide. So here, this top path is SP1's path. 1 nucleotide is getting mutated here. 1 nucleotide is getting mutated in SP2. So total 2 nucleotides have been mutated thus far after 10 million years. Now, another 10, which now we're reaching 20, within this 10 million year time frame, another one gets mutated and another one. And then within this next 10 million years, another one and another one gets mutated. So a total of 30 million years have passed since SP1 and SP2. Now remember, each pathway moves independently from each other. But every 10 million years, each of those organisms is going to mutate 1 nucleotide. So it goes 2 at a time. So again, let's just look at this example again. You have a common ancestor. Here's path 1 right here. This is path 1. Here's path number 2. Okay, every 25 million years, we have a mutation. Oh, look, it mutates A. Versus this mutation here, oh, look, it's mutating C. Okay, the next one, the A has already been mutated. Oh, look, G is now the new one. Oh, C was my original one over here. Oh, look, another C. So that's 2. Okay, so now we have 4 different mutations in comparison to the common ancestor. Okay, so hence why if we have 6 nucleotides, 3 are going to be mutated in SP1, and 3 is going to be mutated in SP2. Okay, so always, however many different organisms you have, split them off into their different pathways. If there's 3 organisms that we're comparing to, then do 3 pathways and just draw a line through all pathways to represent how many mutations occur. Again, how I drew a line is right here. Boom. That's why this is 10. I drew a line. 1 1 1 1 1 1 6 total. Okay, so about 30 million years ago. This is a long lecture, I know. Don't worry. How does analysis of DNA and protein support other evidence of evolution? Again, chapter 11, chapter 8, chapter 9. Okay. So as we've already seen, this cave salamander has no eyes, the snake has no limbs, and the salamander is slightly different from the snake due to the environment. So how might the loss of limbs enhance the fitness of burrowing animals such as a snake? Think about it. If you don't have extra limbs that are bulky, is it going to be easier for you to burrow into the ground, or is it going to be harder for you to burrow into the ground? Okay. And that is it. So hopefully you guys got 51 minutes of straight lecture audio. Hopefully the audio is working this time. If the audio was not working, please let me know so I can fix it. I'm going to do the re-recording of, what do you call it, of last chapter, chapter 12, on the spots that the sound went out. So be on the lookout for that. I'm hoping to get it done by tonight or tomorrow. I'm hoping to get everything done by tomorrow soon. I will be posting up the Canvas discussion board for you guys to do the flex points in. Again, 4 chapters, 4 questions. Label each question to the according chapter. Animal development, just type in AD, so that's all right, so I can tell. Again, concise it all into one discussion post so I can see. I will have a participation quiz set up very soon. You guys can take it as many times as you want, but again, it's based on our lecture, and it's a good way to test your guys' knowledge on what we've talked about. And this is kind of like a study guide practice. It's not an official thing. Again, this is worth 2 points per chapter, so 8 points total for you guys, and it should be easy points for you guys. So that's going to be due towards at the end, the day of the exam. Okay, so I'm gonna hopefully get those set of questions up soon, and then by the day of the exam, it should be up. And for those of you who are in the future, we might not have that anymore. So if we don't have it anymore, don't worry about it. It's probably not going to be on it, and if it's still on it, then there you go. All right. So best of luck. Stay safe. Stay healthy, and see you guys next week.